Hello, it is my great pleasure to present today about the COVID-19 testing and genome analysis. I'm Min-Sup Lee, Chairman and CEO of the Diagnostics USA and EDGC Korea. It has been already 10 months since the first pneumonia patient was identified in Wuhan, China, that caused by a novel virus called SARS-CoV-2 or COVID-19. There are more than 33 million cases of infection and 1 million deaths reported as of now. When we look back the early timeline of the pandemic of the new virus, the most important discovery was made on January 7th with the completion of the genome sequencing of the new virus from the first patient. It took only 2 weeks to publish the virus genome sequence from the sample collected from the patient. Left diagram is the first paper published from the genome sequencing from the virus on January 29th by the analysis of the virus genome sequencing data. The scientists found out that the virus was the new strain of coronavirus with a sequence size of about 30,000 nucleotides. It is a positive sense single-strand RNA virus and believed to be a zoonotic origin from the closely related to the bat and pangolin. The virus is composed of a dozen genes, including 4 structural proteins such as spike, membrane, envelope, and nucleocapsid. They figured it out that the virus mainly enters the human cell by binding to the receptor angiotensin converting enzyme 2, known as ACE2, that mostly abundant expressed in lung by binding to the membrane spike protein. The striking difference of SARS-CoV-2 compared to other coronavirus was the very high rate of infectivity that was estimated 3 to 6, which means that each patient can spread to 3 to 6 additional people. Further sequencing comparison of the new virus revealed that it is the most closely related to the SARS-CoV-1 that was first identified in 2002, affecting more than 8,000 people with 10% mortality rate. In order to further understand the dramatic increase of the infection rate of SARS-CoV-2, scientists compared to the sequence of SARS-CoV-1, and they found that several interesting variation regions, including receptor binding domain and the junction between spike 1 and spike 2 genes that cause the cleavage of the proteins. The most striking difference was found in the junction between S1 and S2 subunit. The junction was known to be cleavage site for the S1 and S2 protein to promote the fusion of ST protein to human cell membrane. There was a unique additional 5 amino acid, which is a 15 nucleotide insertion only found in SARS-CoV-2 compared to other known coronavirus. The site was known to be a furin cleavage site that mediated the cleavage of S1 and S2 subunit by serine proteinase transmembrane 2 enzyme. Scientists estimate that the increasing reproducibility rate of the COVID-2 is most likely due to the enhanced cleavage of S1 protein by introducing a novel furin cleavage site. Some scientists raise a question about the origin of the furin cleavage site and claim that SARS-CoV-2 is most likely introduced artificially by human. Therefore, the virus is most likely man or lab-made virus. They claim that the chance of inserting a 15 base of the new sequence by nature is almost impossible. I do not want to make any conclusion on this, but I leave it up to you for what you can think about this. The immediate contribution of the virus genome sequence information was the development of the SARS-CoV-2 diagnostic testing, particularly molecular or reverse transcription or real-time PCR assays. There are 3 big categories of testing available for different purpose and utility. First, virus molecular test, also known as RT-PCR, is to detect and diagnose the active coronavirus infection by taking nasal, swab, or saliva samples from a patient. The molecular test is highly sensitive and accurate, but it requires a laboratory and multiple instruments to use it. Turnaround time could take several days. On the other hand, the virus antigen test also targets the presence of the active coronavirus infection using a nasal swab. It can be processed very quickly, but sensitivity of the detection is much lower than standard molecular PCR test. The antibody test is the most used for different purpose than the molecular PCR or virus antigen test. The antibody test result indicate whether you have been infected in the past to assess the prior infection of the virus. The positive antibody test result could implicate that the patient carries an antibody that can protect the future COVID-19 infection. There are 5 different types of antibodies present, but 2 antibody testing, IgM and IgG, are most widely used in the clinical assessment. In order to understand the utility of the different type of coronavirus testing, it is important to understand the detection time curve of each biomarkers after the infection. The blue line represents the presence of the virus mRNA after infection. It is estimated that the RNA could be detected up to 3 weeks, with the maximum level around week 2. The virus antigen can be also detected similar pattern as RNA, but with less sensitivity and specificity. The COVID antibody IgM is one of the first antibodies that responds to the initial infection and detectable for about 2 weeks. The presence of the IgM could indicate that COVID-19 immune response is actively fighting against the infections. The antibody IgG is released slowly but lasts very long to provide a longer protection and memory of the future virus infection. Therefore, both molecular and antibody testings are required to accurately assess the patient infection and the recovery status for prevention, protection, and treatment of the patient. This diagram illustrates how to develop molecular diagnostic tests for the SARS-CoV-2. The first target gene, together with the amplification primer and detection probes, are selected by comparing nucleotide sequence information compared to other genome sequence from the database. It is required to optimize the primers and reagents in both in silico and wet lab levels to improve the test performance. Multiple iterations of the cycle may be necessary to develop the robust assays. The right diagram shows the example of the primers and probe designed for SARS-CoV-2 detection. This diagram illustrates the COVID-19 testing process from the sample collection to the result reporting. First, appropriate samples are collected from the nasal or throat swabs. The collected sample is transported to a central laboratory stored in the viral transport medium. The individual patient samples in a tube is allocated in a multi-well plate. The real-time detection of RT-PCR by fluorescent color detection probe is performed after the RNA extraction, followed by lysis, wasing, and elution steps. The RT-PCR result is reported from the CT curve analysis. The testing is normally performed in biosafety laboratory. This slide shows the example of the first few companies in Korea and US that was approved by FDA for emergency use. Interestingly, Korea is one of the first countries that molecular diagnostic test was approved by the FDA. The early approval and successful commercialization of the test lead a very successful quarantine result of the virus pandemic after the first breakout in Korea. The picture shows the diplex COVID detection diagnostic kit from the Solgent, which is one of the first companies got EUA approval from Korean FDA. The importance of the COVID-2 antibody testing is more emphasized as the pandemic reaches to the peak of the infection. The presence of the antibody is very important for the measuring the herd immunity as well as the prevention of the spread. Furthermore, immunization by the vaccine can be monitored and measured by the antibody testing. This slide shows the two different types of antibody tests available. The first one is the rapid Cellulosical testing using a lateral flow test strip similar to the pregnancy testings. The result can be detected within 20 minutes and the testing can be performed by the patient. On the other hand, ELISA, or known as enzyme-linked immune assay, analysis normally performs in the reference laboratory. The ELISA test takes several hours and yields quantitative measurement of the antibody and shows much higher sensitivity and specificity compared to the rapid antibody testings. Some of the new products such as EDGC Rapid Check shows in the left picture has the advantage of on-site testing capacity with the quantitative measuring of antibody level within 2 minutes and gives the better sensitivity than standard rapid check testings. This diagram shows the process of both rapid and ELISA testings. The rapid testing can be performed in the simple lateral flow device or strip. The result can be displayed as a visible line together with the control antibody. The ELISA requires multiple steps of sample incubation, antibody binding, and signal development stage. The result can be measured by fluorescent plate reader. The COVID-2 antigen testing could potentially detect the presence of the virus molecule in the infection stage by simple nasal swabs. The Abbott's Binax Now COVID-19 antigen test got the most attention in the media recently, even before the approval of the FDA EUA. The Abbott announced the $5 15-minute rapid antigen test with a sensitivity over 97% and specificity over 98% when the test was performed to the symptomatic patient within 7 days after symptoms. The test is approved for the use of point-of-care settings with the specimen collection performed by a healthcare professional under clear requirement. The Trump administration pre-ordered 150 million rapid tests and planned to deploy it to the state for the most school and work. So it is most likely that this rapid antigen testing is only available in the United States at least for the next 6 months. In addition to the standard molecular RT-PCR test, that can only detect the COVID-19 virus infection status, the genome sequencing of virus extracted from the infected patient gives another level of traceability and classifications. The virus genome sequencing can be performed using next-generation sequencing such as Illumina NextSeq or NovaSeq machines. The steps include RNA extraction, cDNA preparation, RT-PCR, target amplification, and sequencing library preparation and the parent sequencings. The sequencing result can be analyzed and assembly and alignment to a virus reference sequencing following by the mutation detection. There are several bioinformatic tools are also available in the Base-based Genome App Store to analyze and annotate the sequencing data to find the mutation and assign clade or subtype of the virus. This is an analysis result of sequencing 48,000 viruses from the patient over 100 different countries. The sequencing data has been voluntarily deposited to a public virus genome database named GSAID. Meta-analysis of the data revealed that the average number of the mutations found in each patient genome was 7.3, ranging from 0 to 70 mutations when the individual sequence was compared to the reference genomes. Over 13,000 different mutations have been identified from the collection of sequencing results, indicating that the mutation could happen almost every position of the genome in the virus. There are about average 10 difference was found when the sequence was compared to the two random peoples. The right table shows the 20 most frequent mutations observed in the virus genome database. The first 4 mutations, including the position D615G in the spike protein, were mostly found together, indicating that they are linked to each other from single origins. Recently, Korean CDC announced the subtyping of clade information of the patient based on incidence location from the sequencing patient's virus genomes. Most of the recent outbreak, including Itaewon, is categorized to type GH compared to an initial outbreak in Korea, mostly Shincheonji Church, that was known to be a type V. The sequencing and classification of the virus can not only group important subtype of virus, but also can be used to tracing the virus for the transmission and spread. By comparing the difference of the mutation in every genomes, you can trace and track the origin of the infection and the subsequent spread of the chain infections. The clade means branch in ancient Greek, meaning that composed of the common ancestors. The left diagram illustrates the standard way of classifying the SARS-CoV-2 virus substrain based on the phylogenetic tree analysis for clade separation. There are several different ways of doing the classification, but the GSH classification method is most widely used in grouping the COVID virus. The classification is made by the analysis of the common mutation found in virus as shown in the right tables. This picture shows the global distribution of the different clade of the virus. The left map shows the distribution of the clade by countries as of September. The blue, the G including GR and GH clade are the most prevalent than any other clade. The right chart shows the distribution of clade by each countries expressed by a different time point. L strain is the original strain of the COVID-19 that originated from Wuhan. The S strain was the first mutation of the original L strain of the virus, mainly found in Asia. V strain is about 7% of class of the worldwide, most of which are in Europe. Strain G and its related strain GR and GH are by far the most widespread strains and are most often found in South America and Europe. The GR strain carries a combination of the spike and nucleocapsid protein mutation and believes the faster spread than any other viruses. And the clade GH is most abundant in North America. This is another view of phylogenetic trace information based on GSAID and x-axis on the date of the infections. Again, it's clearly shown that G strain prevails any other strain observed as of now. This is another view of phylogenetic trace information based on countries plotted x-axis on the date of infections. The red color includes both Korea and Japan. You can clearly see the power of tracing each individual genomes based on genetic clade classifications. This is the distribution of the mutation throughout the COVID-19 genomes. There are over 13,000 different mutation positions out of 30,000 whole genome lengths. As you can clearly see in the left diagram, the individual variation frequency has been dramatically changing in two different time points, May and September. The striking difference in this distribution in different time points indicating that viruses are under strong force of evolutionary pressures. Furthermore, the right diagram shows that distribution of mutation found in four structural proteins, spike, envelope, membrane, and nuclear capsid, that are most important in the transmission and characteristic of the virus. The red dot is a non-synonymous amino acid changing mutation that could potentially alter the virus infectivity and antigenicity. The blue dots are the silence mutation that does not change by any amino acid. Surprisingly, spike proteins are under high mutation burden compared to other areas of the structural genes. This is very alarming that many of the vaccines that are under development may not work in some of the people because of the mutation present in the spike proteins. Most of the subunit vaccines are targeting the spike protein for the vaccine development. Therefore, it is also very difficult to predict the future mutation because unexpected new dominant mutation could arise from any point of the genomes. Therefore, it is very obvious that the multi-target approach for the vaccine target selection is very important for the effective and long-lasting the vaccine development. Among the many other mutations known, the D614G mutation in the spike protein is the most widely found mutation in the class G, including GH and GR clades. The mutation changed the amino acid position at 614 of the spike protein near the junction of the S1 and S2 protein subunit from asparagine to glycines. The D614G is known to increase the infectivity of the COVID-19 virus. It is estimated that the mutation is up to 8 times higher reproducible rate than original L or F types clade. It is speculated that the D614G mutation may confer a competitive advantage at the furin binding domain. As you can see in the global transition in the top of the right pictures, most of the new virus instance is from G recently. The old D form is almost extinct now, meaning that we are in a stage of second pandemic that is mostly attributed by the D614G mutations. This is the summary of my presentation. Instead of going through all the line by line, the take-home lesson is that the genome sequencing effort really contribute the better understanding of the nature of the virus and provide a valuable insight for us to deal with the pandemics. And based on all these genomics researches, we are able to provide the right testings and tracings and even the right treatment for the patient. Thank you very much for your attention. That will conclude all my talk with great thank you to all of you. I'm open for any questions and suggestions. Thank you.